continue joining. Hi, everyone. Welcome to the University of Washington Division of Rheumatology Grand Rounds. A couple of reminders before we start the session. This session is currently being recorded and it will be posted later on our website. There will be a Q&A session at the end, so please put your questions in the Q&A feature below and not in the chat box so it's easier to moderate. And now I'll let Dr. Mustelin introduce our speaker for today.
Thank you, Danielle, and good morning to everyone. It is a real pleasure for me today to introduce our guest speaker, Dr. Timothy Niewold. Dr. Niewold got his MD degree in 2001 from the University of Illinois at Chicago. He completed an internal medicine residency at the Mayo Clinic in 2004, and then a fellowship in rheumatology at the Hospital for Special Surgery in New York in 2007. He then went on to serve as instructor of medicine at the University of Chicago for a couple of years, then became an assistant professor in 2009. In 2012, he accepted an associate professor position at the Mayo Clinic. And then in 2017, he moved to New York to get an endowed professorship in medicine and pathology at New York University. And then in 2022, he became the vice chair of medicine for research at the Hospital for Special Surgery. I know that he earlier overlapped with Keith Elcon while he was at the Hospital for Special Surgery in his first time. Dr. Niewold has gone on to numerous leadership positions. He is very active in several organizations that support lupus research and has trained a very large number of trainees and fellows and postdocs. He has well over 200 publications. He is extremely well funded, and I think most known in the lupus community for his work on interferons and lupus and a number of other molecules and related diseases. And I'm going to turn over to Dr. Niewold to present for us today about type 1 interferon. So please go ahead.
Okay, great. Thank you for the kind introduction, and thank you for having me here with all of you, many colleagues and friends at University of Washington. So I'm very happy to be able to speak with you this morning. Let's see, to advance, there we go. These are my disclosures, none of which are directly pertinent to this talk. So yes, I'll start with kind of basics type 1 interferon. The type 1 interferons are important for our viral defense, really important in almost all multicellular organisms, actually, for viral defense. So it's really an ancient defense mechanism. And the best known members of the type 1 family include interferon alpha and beta, but there are also some lesser known members such as interferon lambda and interferon kappa. They all signal through the same receptor. So in the cartoon, that same receptor can actually mediate signals from these, except for interferon lambda has a separate receptor, but that's a type 3. But interferon kappa, for example, signals through the type 1 receptor. And as early as the 1970s, we've known that levels of type 1 interferon were detected in lupus. And this was using a functional reporter cell assay at the time. Interferons got their name from their ability to interfere with viral infections before we really knew exactly what the molecules were. If there were supernates that contained these interferons, you could put them on cells that had been infected with virus, and you could see that it would slow viral replication in those cells. So this was a discovery from quite a while ago, actually, in 1979. So we knew that high levels were found in patients, but there's still, this is always hard to determine in humans. Is there evidence for causation versus association? Is it that someone gets sick with lupus and then later on interferon becomes important, or is it something that could precede or even cause disease? So I think with type 1 interferons, there's a really interesting story and a number of different leads that suggest that interferon is at the earlier part of pathogenesis and that it might be causal. So one of them, this is how I got interested in interferons and lupus in the first place. I was a medical student, and I had seen a case of a patient who had gotten recombinant interferon alpha to treat hepatitis C, and she developed a pretty classical case of lupus, actually. So she had malar rash and double-strand DNA antibodies, and really some very classic features of lupus. And when we took away the interferons and gave a short course of steroids, she got better. Some of the ANA actually persisted, but her clinical symptoms got better and didn't come back. And there are a number of cases in the literature. When I had seen this case, this had already been reported a number of times, and I remember looking it up and realizing that this had been a shared experience, and really fascinating that in some people, the addition of interferon alpha was enough to push them into this complex disease, lupus, and then almost like Koch's postulates, then you remove it and the lupus goes away. So pretty striking finding. We wanted to explore this idea a little bit more. How can you make inference about causation in humans? And so started wondering if this might be something that could be inherited. And we did at the start of sort of an old-fashioned heritability study where we looked at lupus patients and their family members and measured interferon levels across the whole extended family. So first-degree relatives like mothers, fathers, brothers, sisters, and then we luckily had many grandparents, uncles, aunts, and some people that were farther removed. And we did find in the first-degree relatives echo of the high interferon that you see in patients. So in this graph, the patients obviously have the highest levels, but you could see some abnormally high levels in all the different categories of first-degree relatives. So mothers and fathers, brothers and sisters were also very similar, so they're lumped together as siblings. And if you measured healthy unrelated people, the average is lower. And so interesting, in human studies, there are always potential confounders. The lupus patients were generally adults, and so they were tended to live separately from their family, because you could wonder if maybe a virus runs through the family, then you could get a correlation in interferon levels related to a shared infection rather than anything else. But these were tended to be samples collected at different points in time, and many times the relatives actually had to mail in samples to this registry. The other way that we tried to control it is we measured interferons in spouses of lupus patients. So I don't show this on the graph here, but we looked at a number of spouses, and they were similar to healthy unrelateds actually in their interferon levels. So that would be a control in that you have someone sharing a close environment with the patient, and yet having a different interferon levels, so to try to help exclude environmental causes. Again, nothing's perfect, so many more of the spouses were male than female, because many of the patients were female, and that was the most common married couple type that we studied, but it was a useful control, at least. So around this time, actually, there was a lot of genetics work going on, and the first GWASs were going to come very shortly thereafter. And one of the interesting things, if you look at genome-wide association studies, which are sort of an unbiased sweep across the genome to see which genetic variants are associated with disease, quite a bit comes up in the type 1 interferon pathway. Also some other inflammatory pathways like NF-kappa B, and then toll-like receptor signaling, and some of the downstream interferon components come up as well as genes in which there's a variant either within or very nearby that gene that are associated with risk of disease. So I think this is another line of evidence that helps support the idea that interferon is causal in disease. I think with candidate gene studies, it's hard to say, because with candidate gene studies, you just pick what you want to study and you find it or you don't. So it starts off with a bias, but even when we get to these kind of more unbiased genome-wide screens, interferon comes up much more often than it should by chance alone. And so we've gone on to study this. Given a family study, we were interested in seeing if you could show that some of these risk alleles, when carried by a patient, were associated with higher interferon levels or not, to try to kind of do the test in people. And we did find a number of things that were associated. So, and it seemed to follow, I guess, a similar rule that many complex genetic diseases follow. We didn't find a single one gene that was explaining the interferon, but we didn't expect that, actually. So in the family study, our pattern mostly fit a complex inheritance pattern in which the first-degree relatives shared the trait. When you got to second and third-degree relatives, the co-occurrence of the trait dropped dramatically. And so that's typical of complex disease, is that the risk of that disease drops logarithmically by degree of relation, whereas single gene defects, it's much more of a linear drop in risk. So we saw a number of common alleles that had a modest effect on interferon. We did see, I'll present this middle circle, the IRF5 with the autoantibody interaction. We did find we've been able to find some interactions where you see a sort of a multiplier effect. And then in work by many other groups, you've been able to see the sort of rare allele with large effect phenomena as well, if you are willing to count the interferonopathies and some of the lupus-like diseases you can get from some of these rare variations that cause high interferon levels, and some lupus-like phenomena such as Chilblain lupus and the interferonopathy spectrum. So I think you can make this graph suggesting that this is a complex trait largely, but then there are also some single gene variants that can cause a similar phenotype. And I think that's generally what we've seen in a lot of our autoimmune rheumatic diseases, that there are kind of both conditions. If you think about the disease as a whole, if you think of lupus as a whole, we have a lot of common alleles, and then we also have some monogenic variants that can lead to a similar phenotype. So just in a summary cartoon, we found a number of genes along different points in the pathway that all seem to fit sort of a gain of function model. So we had binned those largely into things that were upstream of interferon and tended to be downstream of toll-like receptor signaling that resulted in a higher level of interferon in the circulation, is how we would usually measure it. It's, we can't get tissue levels very easily on all kinds of different tissues, but the blood's very accessible, and as probably many or all of you know, that the interferon signature in blood is pretty strong in lupus patients, so we can get at least that whole blood assessment of interferon in patients. We did see some polymorphism. For example, this is STAT4 in particular, downstream of the type 1 interferon receptor, where we saw what we call the sensitivity phenomena, where when you carried the genetic variation for a given level of interferon, you actually got more downstream signaling as measured by the induction of interferon-induced genes. So that was interesting, and we felt like you could sort of make a case for both pathway locations, both the increased production of interferon and an increased sensitivity through some of the downstream sensing mechanisms. And that would make sense for a complex disease, and just in general, that Mother Nature's clever and can think of many different ways to modulate a pathway. I'm just going to show you one story in a little more detail. This has been very overview, but we had some studies with IRF5 that I think were interesting and illustrative of some of the human heritability and how these things come about. So we thought IRF5 would be a slam dunk. This was a gene that was found before the GWAS studies even. This was one that people were very interested in, including the other interferon regulatory factors, of course. And when thinking of which genes might lead to higher interferon levels, this was a logical choice because, as the name suggests, these are transcription factors that can induce interferon genes themselves, as well as a number of interferon-induced genes when they are activated. So seeing a risk variant here, we thought this should be part of this familial interferon thing that we're seeing. And when we studied it, we could see kind of, but not, it wasn't simple. So when we first looked with all patients together, the effect was very weak. And even studying many more patients, you could see a trend, but it was pretty weak. What made a difference is if we looked by autoantibody subgroups and genotype. And so that's what these graphs are showing here. So in graph A, these are patients who either lacked double-strand DNA or had double-strand DNA antibodies. And then the 0, 1, and 2 on the x-axis is carrying either none of the risk haplotypes, 1, or being homozygous for the risk haplotype. And so we didn't see anything in the patients who lacked autoantibodies, at least the double-strand DNA antibodies, and that was about half of the cohort that we studied. But in the other half, we saw what we were kind of expecting to see, is a stair-step increase in circulating interferons by increasing carriage of the risk haplotype. When we split by anti-Rho, this was also very similar. So this is European ancestry, and these were our two most common autoantibody types. There was some Smith and RMP, but as you know, the numbers of European subjects who carry those autoantibody specificities is much less. But you could see a similar phenomenon where you could see the genetic effect in the autoantibody positive subset. So So it's interesting. So that seemed like the autoantibodies were having a mediating effect on this genetic impact on interferon. And I thought that could make sense if we go back to the previous diagram, that maybe you need the autoantibody immune complexes to stimulate the system to really then, sort of see an observable genetic effect in the circulation. Maybe it takes quite a bit to dysregulate your whole body circulating interferons, and maybe this autoantibody sort of constant endogenous stimulus is needed to bring out the genetic effect. So when we thought about that, we thought we could try to test this in some way. I guess test as a geneticist would test it, and we thought, let's take a look at the frequency of the genetic variants. Is a question would be, if interferon is what's causing lupus, and there's one group of patients where the gene makes a difference on interferon and another group of patients where it doesn't, we should actually probably see that the gene is enriched in these people who in which interferon is actually modulated. And maybe it doesn't matter so much whether you carry or don't carry that haplotype if you don't have the autoantibodies. And so when we went back to the genetic data, that's what it looked like, actually, is that there was more than 70% of the genetic risk of lupus related to IRF5 was within these serologic subgroups, which again was around a little bit over 50% of our cohort, I think, that had either one or both of these autoantibody specificities. And so, so that was interesting, and a sort of a major subgroup effect that we wouldn't have detected by our overall GWAS. The overall genetic studies, by and large, group together as many patients as you can get of all different types, and then try to study everybody together to increase statistical power. But there's always this chance, I think, that there are subsets running around in the data. It's just hard to know, hard to know what to look at and why, and how to subset patients to pull them out. So that was interesting, and it's not 100%, but I think that that's probably expected. Nothing's 100%, but also you could imagine that there would be other stimulators of this pathway besides just the autoantibody complexes that could be sort of upstream of IRF5 and cause that variant to maybe take on a particular significance for other reasons. So it really shouldn't be just autoantibodies, but that would seem to be a fairly major effect with regard to this gene variant. So we studied multiple populations as well, and one of the interesting things is that we found it at pretty high frequency in Europeans, not nearly as high in African Americans. And when we went to African populations, so native South African populations, we didn't find the risk haplotype at all. So to orient you, the first one here, this TACA, is the best described risk haplotype that we were showing in some of the graphs before. And when it's present in African Americans, it's present at about 20% the frequency of European Americans. And so one hypothesis that jumps out right away, if you see that, is that it may be present just due to admixture. And that's when you find 0% frequency in native African populations, that's the most likely thing, actually, because the average European admixture in African American populations in the US is around 20%. So when we studied interferons in the African American patients, we saw a similar phenomenon. Because the haplotype is so rare, we didn't have many homozygotes to make the 0, 1, 2 categories. We kind of had to lump together 1 and 2 together, but you could still see some trend toward an increase only in the autoantibody positive subset. So it seems like it might be functioning similarly, but it was interesting that it seemed to be of European origin potentially. So that was an interesting question and a puzzle for us. We thought about this for a while, and nothing really made a lot of sense. We had to just go ahead and just publish what we had because we couldn't come up with any real good hypothesis about this. It's all of us originate eventually from Africa if we trace our roots back to our origin of humans. And after that, we've spread across the planet, in different lineages. And each one of these lineages would be expected to carry some fraction of the overall diversity that's found in Africa. And so in Africa, there's the greatest amount of human diversity. And as you get farther away, there tends to be less because these places were established by some founder population that carried with it some, but not all, of the genetic variation present in our original population. So we could hypothesize maybe this variant was evolved de novo in European populations, although it's pretty, that'd be weird. And it was a long haplotype. It wasn't just one single genetic variant. It was a long haplotype with a couple different functional variations. Could hypothesize it went extinct in Africa. That's also probably unlikely unless there was some overall strong genetic selective pressure. And then after that, we really didn't we really didn't know. So we left it there for a while. And as usual, the truth is stranger than what you can dream up. And when this paper came out, we got our answer. So it was one of my friends in genetics who called me and asked if I had read this paper, and I said no. I said I saw the news and views, and it's interesting that they made the Neanderthal genome. And they said, well, you should read it. There's lupus in there. And I said, really? I said, I didn't think that's strange. I didn't read it because I didn't think there would be lupus in there. But he said, no, you got to check it out. He's like, there's one of the lupus risk genes in the Neanderthal genome. And it turns out this is the one we were studying. So this was this haplotype in lupus, and it's actually really pretty common in modern day European populations and also other populations that have Neanderthal admixture. And it's even noted, this is from the original paper, that this was one of the phenotypes along with primary biliary cirrhosis that the haplotype had been associated with. The particular marker that they got was different than ours, but they were in basically 100% linkage equilibrium when we looked with this long risk haplotype that was present in European populations. So the Neanderthal range was really from Europe and into the Caucasus. There is some Neanderthal ancestry as you go all the way into Asia, likely transmitted along the Silk Route, but really none in sub-Saharan Africa. And so this is I think our explanation of where this haplotype came from and why we didn't see it in African populations. And very interesting, I think, that this is something that was carried forward. So the Neanderthal genome, as you'd expect, is not equally represented and sort of, almost like a random distribution across our genome. There are certain things that are more enriched and certain things that seem to have dropped away, likely due to some recent human selection. And I guess to put it more simply, we probably kept some of the things that were useful to us and let go of some of the other things that weren't useful, or especially anything that was not productive or caused any selective disadvantage. Interesting. It's not that generalized of a phenomenon. So if you look at other lupus risk alleles, it's not that all of our risk of lupus came from Neanderthals by any means, but there are a number of instances of innate immune system genes that have been carried forward from other hominid populations. So there are some toll-like receptor gene variants that have been traced back to Denisovans populations, which were more of a Central Asian hominid population that was I think in time fairly similar in time frame to the Neanderthals. So taking a step back, and trying to put these things together, we have a gene from Neanderthals seems to interact with autoantibodies, resulting in higher interferon. To think about, how lupus occurs and what we know about pre-disease, there's obviously the very famous studies by Judith James and the Oklahoma group looking at pre-disease serum samples. And so the autoantibodies, as probably everyone knows, to come up to, even 10 years before disease diagnosis or classification, we were able to test interferon, type 1 interferon in these samples. And their group actually tested type 2 interferons as well, or interferon gamma. And very interestingly, the interferon gamma levels came up even a little bit earlier than the autoantibodies. And a number of autoantibody specificities, these are the different multicolored lines here, come up again years before disease classification. Type 1 interferon we saw kind of shoots up within about 2 years before disease classification. So I think it's interesting, it sort of fits into our potentially this kind of two-hit model that we've wondered about. Maybe these genetic variants, if you have an IRF5 allele, risk allele, but you don't have the autoantibodies, maybe that's fine. And maybe it's there waiting in case you get a viral infection. And then maybe you're one of the people in the office that doesn't get the cold when it comes through, and maybe you are one of the lucky ones that doesn't tend to get viral infections as much because maybe you've got some moderately, increased either production or sensitivity to make type 1 interferon-induced responses. So this could be beneficial, maybe why we retain the Neanderthal allele and also maybe why a number of the lupus risk alleles are fairly common actually in the population. And then in this model, you could wonder if maybe this formation of autoantibodies that can stimulate the toll-like receptors via immune complexes might be the critical event where then you have this chronic endogenous stimulus. And if your pathway is already tuned to be hyperactive, then maybe this is where you sort of head down the road of no return, where you get a tolerance is already broken, and then you can continue to spread, epitopes around your already autoimmune response. and then you'd have interferon, which would turn on a number of other immune mediators down the line, and then maybe this is where it becomes more irreversible. And of course, as we know, the formation of these antibodies is actually pretty rare in the general population. It's uncommon to find most of these autoantibodies outside of lupus patients or other autoimmune affected patients, and so much so we can use them as a diagnostic marker. Okay, so further along this story, we had always wondered about how we could test this, the other side of the coin, thinking about, are these alleles actually helpful? We can theorize, geneticists like to theorize, oh, that must be what it is, and maybe, but, it's always nice to test things too. So we got our chance. We were wondering about hepatitis C, but we could never really line up the right cohorts to test and study. But with COVID, we actually got a pretty good chance to study this. So, and there were some papers actually that came out before we did our study, fairly early in the pandemic, showing that either severe genetic defects like monogenic loss of function, severe loss of function type 1 interferon pathway, or if you made neutralizing anti-interferon antibodies, you were more likely to have severe outcomes or death after COVID-19 infection. So we wondered about the common allele hypothesis, if we could look at some of these common lupus risk alleles that modulate interferon, and if you could see if they were associated with protection from severe outcomes. So we had access to genetic data. We didn't have serum samples, unfortunately, so we couldn't test the actual interferon levels, but we had access to genetic data from a first wave New York City cohort where there unfortunately were quite a few severe outcomes, and so it was reasonably powered for detecting, associations with severe outcomes. And we were able to look at a number of risk alleles. We tried to put in many of the risk alleles that we knew were associated with higher interferon levels, and a number of them came out significant. So the first risk factor here, this TCTA, that's one of the IRF5 haplotypes. We also noted IRF7, which is this RS702, and actually the one below it, so two different polymorphisms in IRF7. IRF8 is right above it. And then actually this PRKG1 allele that we had discovered in one of our interferon screens was also one of the strongest associations. And the graph here is just kind of a cartoon, actually. So the new data we generated was the COVID-19 mortality odds ratio if you were carried any one of these polymorphisms. And then we took data from our lupus patients to compare. So this is not from the COVID-19 infected patients, but this is the ratio of interferon you get whether you either carry or don't carry that particular risk allele, and how much of a fold change it makes in your interferon levels as a lupus patient. But even though it's derived from different populations, it was interesting that you could see a general correlation between COVID mortality odds. So the higher interferon polymorphisms tended to associate with lower COVID-19 mortality ratio, and then opposite for the ones that were associated with lower interferon levels. So that was exciting. I know there have been a number of other studies that I think have generally supported this idea now as well. So again, thinking about, this is also leading another study that we've done in relation to viral infection. We've known that type 1 interferon levels vary across different ancestral groups. And it's pretty marked, actually, when you look across major world populations. And that's the first graph over here to the left. European ancestry subjects tend to have lower interferon levels. This is all lupus patients. So this is European ancestry lupus patients have lower levels than generally other African American, Hispanic American, or Asian American populations. And the others, these are all fairly similar. And so we thought that was interesting in a number of ways. I guess one, in that European ancestry tends to be the least commonly and least severely affected ancestral group, and also tends to, if you do get lupus, actually tends to have lower interferon levels. We had done some studies with a number of European collaborators, and one of the things that struck us was this middle graph. So we had done studies with the Karolinska and noted similar, even maybe slightly higher interferon levels in their group than our American group. And then we did studies in collaboration with Crete, and we noted lower interferon levels on average. And it was interesting, speaking to the lupusologists in Crete, that they said, yeah, we read about the interferon signature and we see it sometimes, but it doesn't seem like that big a deal to us. They said, I don't know, maybe it's probably there, but I don't know. We see a lot of patients don't have it, and so it was interesting. They wanted to try to publish something right away, and I said, I don't know if we're going to get that across the finish line, because we're going to get endless, complaints about like maybe your freezer's failed, or maybe, I don't, who knows, maybe there's environmental other things, it's going to be hard to make any kind of a decent paper with just that. So we decided to study our European American subjects, but by looking at their relative contributions from Northern and Southern European ancestry. So there are a number of genetic markers you can use to sort of understand whether someone has more Northern or Southern European ancestry. If you've seen the, uh, at-home genetics providers, that'll tell you your own ancestry components. The commercials, say, oh, I didn't know I was Swedish or whatever. Anyway, you can get fairly fine point on that, actually, as far as determining where your recent European ancestry is from. And so we thought if we study patients here in the US. So again, this is still lupus patients, but study the patients and see, are they more Northern or Southern European derived genetically? That's kind of a nice control, because then all the chromosomes are within the same person. And if their interferon level is different, then that's a much more, I think, striking corroboration of the data than by just studying different locations, which always have geographic confounding. So that was what we found, basically, is if you had higher European ancestry, that's in panel B, Northern European ancestry, you had higher interferon levels as a lupus patient. And if you had more Southern European ancestry, then you had lower interferon levels as a lupus patient. So we felt like this might be a general trend. And so we studied some of the known European populations samples that are available. This is from the HapMap and 1,000 Genomes project, and did some measurements, but also inferred some of the likely interferon scores in the different populations. This is healthy people now. This is switching to healthy people that were available through the various genomic consortia. And so you could see a similar thing where the graph shows the quantitative data, and then there's sort of a European heat map type of diagram on the right, where red would be higher interferon, yellow intermediate, and green lower levels. And so we saw between Northern Europe, far northern more so, and then a little bit lower in Western Europe, and then in Central and Southern Europe we saw lower interferon levels and predicted interferon levels. And so we were able to get sort of the final part of this paper, we were able to get some of the population level outcomes data from a number of different viral infections. So not every country had it, but the countries that did have, we were able to analyze it. And it was interesting in that you saw a similar sort of inverse correlation if you were this is at the population level, of course, but if it was a country that was predicted to have on average higher interferon levels, they tended to have improved viral outcomes as measured by death per case in COVID or death per case in hepatitis B. Some confounders can't be overcome in this, of course, because there are differences in healthcare, differences in the sort of wealth of the nation and how well they're able to respond to COVID-19 or hepatitis B. And so, these, so I would say this still is suggestive, but I think interesting. And I think we feel like there's probably some contribution, at least, to these sort of background genetics of interferons in these populations and their ability to respond to viral infections. Okay. So summary of the first half, or probably more than half, type 1 interferon levels in lupus patients are impacted by autoantibodies for sure, but also common genetic polymorphisms. And then an IRF5, we called this sort of a gene microenvironment interaction, where you could see this interaction upon interferon and sort of a non-Homo sapiens origin. This is an ancient allele, actually, that seems to be currently in our risk panel for lupus. And really, I think our background does shape set our stage for cytokine responses, and many things are also critically important, but I think it's one of the factors for sure that sets up how we respond to both autoimmune disease risk and to infection. Okay, so we'll get a little bit more clinical now. I have two pretty short stories here at the end. Clinical disease associations with interferons. This is number of these things have been pretty well known. So younger lupus patients have higher interferon levels. The autoantibodies are pretty important. So patients who have a lot of the clinically tested autoantibodies like anti-Ro or Smith or RMP tend to have higher interferon levels than patients who don't. And ANA is typically not strongly associated. We saw a weak association here, which I think is probably driven by the subserologies. lupus nephritis comes out pretty strongly. And so I'll dig into that a little bit on the next couple slides. SLEDAI score always comes up. It's been very interesting. That seems to be more of a cross-sectional correlation than a longitudinal association, unfortunately. So it hasn't become our interferon hasn't become our blood SLEDAI, unfortunately, because it seems like high patients just stay high and low patients stay low, probably because some of the influences are genetics and autoantibodies and things that don't change very much. Medications too, it's surprising, but this is how it tends to always come out, at least in our hands, and I think in many other people's hands, is that prednisone or steroid use is one of the few things that really seems to have a big impact. And hydroxychloroquine and a number of the other oral agents, at least in large cross-sectional studies, we don't see a big impact of those upon interferon levels, at least in the blood. So for the Renal Disease Association, we were interested in pushing ahead a little bit, and we wanted to see if maybe we could understand this a little bit better and even think about is interferon being produced locally in the kidney potentially. We had known for a little while that plasmacytoid dendritic cells, which is one of the cells we think may be making interferon in lupus. It's a little more controversial lately, and I agree that it should be controversial because I don't think we really know. But it's interesting that if you study PDCs in the circulation, they tend to be lower in lupus patients, and we did a little twist on that where we bin patients into either high or low interferon, and it seemed to be even more emphasized in the patients who had high circulating interferon levels. So one of the ways that we thought, and I think people have hypothesized might be the way this could happen is that maybe when PDCs get activated and go to inflamed tissues, maybe they make a lot of interferon and they're recruited to inflamed tissues. And then if that spills out into the blood, maybe that's why you don't see as many PDCs in blood because they're doing their work in the tissues and then they're not as present in the circulation. So that was sort of a going hypothesis for quite a while. And we tried to look at this in kidney tissue. This is a histology slide where we measured the transcripts for BDCA2, which is a good marker for plasmacytoid dendritic cells. That's in blue. You can see a number of these dark blue dots here that should be plasmacytoid dendritic cells. And then we measured IFIT1, which is a sort of a classical interferon-induced gene. There's a bunch of these little red dots here scattered throughout different cells. And so the idea was that we wanted to see if we could co-localize these things. We thought if there's a group of PDCs like there is in this close-up shot, then there should be maybe a lot of interferon-induced gene expression, and then areas in the renal tissue where there aren't as many PDCs, maybe there won't be as much. And if we could get a correlation, that would be at least correlative evidence that something, there's sort of local production. And turns out we didn't see that at all, actually. So the PDCs, this is a large view, and just to give you sort of the sense of it, these you can see sort of the glomerular formations here, a few really pretty good glomeruli and others that are sort of halfway in and out of the section. And then you can pretty clearly see the tubular structures. So the PDCs were most often aggregated in the tubular structures. You can't really see that very well here. But the heat map grading here is the interferon-induced genes, with blue being low interferon-induced gene expression, and then lighter blue to green and yellow being higher interferon-induced gene expression. So this surprise here was that it was very much glomerular, and that was not where the PDCs were. And it seemed like that was really the sort of fairly simple explanation of where the interferon-induced gene expression was. So we thought we don't know for sure, but it could fit with actually a circulatory or blood source, being that the glomerulus is closest to the blood as compared to the other tubular components. And it could be that it's being stimulated in the glomerulus as the blood passes through. And it didn't seem to support sort of an intrinsic PDC-driven interferon production in the kidney. So, sorry to leave you with a mystery on this one, but I think this is where the field is that you can see high interferon in blood. It's associated with renal disease, and you can see a lot of PDCs in the kidney, but it doesn't seem like they're locally producing it, at least how we tried to test it. And a number of other studies, for example, the AMP consortium, when they look at single cell gene expression in the different cells in the kidney, they really haven't found interferon producing PDCs, at least by transcriptional outputs, that you can't find a PDC cell determined by transcriptional profile that has interferon transcripts in it. So still a little mysterious. I think the questions, we know there's a decent amount of interferon alpha in the circulation for sure. There's also interferon beta, and an interferon alpha versus beta in the tissue, I think we don't know very well. There's probably both, and interferon beta may exert a bigger influence at the tissue level. in part just because many more cell types can make it, and a lot of tissue cell types can make interferon beta. So is this contributing to what we see? And is the interferon alpha we see in circulation more of an overflow phenomenon? And I think in general, the cellular source of this overall interferon signature is still somewhat mysterious. So watch this space. Okay, and then, oh good, I still have some time. So my last short story about interferon and lupus is that we were curious if you use the interferon trait, since it seemed to be pretty stable in patients, if you use this as a way to try to find genes related to the trait. And so we had done a GWAS without any healthy controls, but instead just in patients, binning the patients as either high interferon or low interferon. And we even sort of cut out some of the middle so that there wouldn't be so much of a spectrum, but that we could have two groups that were fairly separate by their interferon in blood. And so we were able to reanalyze the SLEGEN GWAS consortia data as people had serum available, and so we could measure interferon in serum from the same patients that had genetic data. And then we did a replication with the Oklahoma group with a number of patients who were separate from the GWAS and took forward some of the best candidates from the GWAS and replicated those. So we thought, if this is, if interferon is truly a pathogenic subphenotype, there should be some genes that are related to interferon and also some that would relate then directly to disease pathogenesis. We thought this type of analysis might be a little cleaner. You might be able to find genes a little more with either greater statistical power or greater effect size because you're looking at just a part of the disease instead of trying to genetically map the whole complex disease all at once. And I think in general, this is one of the issues, is that different genes are obviously present in different patients. And if all of our complex diseases had, just a few genes that did everything, then we would be done a long time ago. But I think it's unfortunately different cassettes in different patients that end up causing disease. And so when we look at everyone together, the patients who that particular cassette is relevant for will exert a signal, and then the other patients where it's irrelevant sort of dampen that signal immediately in your study. And I think that's one of the reasons we tend to see weak overall associations when we do complex disease studies. So as output from a GWAS study, it ends up being a gene list. It's pretty simple at the end. So we got two things that really passed any kind of threshold for genome-wide significance, a fairly stringent cutoff actually for genome-wide significance. PRKG1, which I won't get into too much, but I'll note it was actually the one of the strong genes in the COVID response. And we've done, I guess, some follow-up too. It looks like it's probably important in EBV control as well. But we've pushed into this PNP gene polymorphism, the second one on the list, more so. We did pick that one off the list because the SNP that was associated was a missense polymorphism as well. So we thought we'd jump in with that. And I guess some of the other reasons we picked it is that there's been some literature on PNP. And so there are known humans who have an autosomal recessive complete deficiency of PNP, and they have an immunodeficiency in some ways. They have a defect in T cell numbers, impaired humoral autoimmunity. They also, interestingly, have been reported to have some autoimmune features, usually hematologic things like autoimmune hemolytic anemia or ITP, but they've frequently had ANA, and there's one case that clinically looked quite a bit like lupus with an ANA and also some arthritis and other things. So PNP is purine nucleoside phosphatase is the long name for it. And the diagram here shows its function. I'll admit this hit some pretty dusty brain cells for me when we found it, but it's part of the nucleotide salvage pathway. So it's somewhat analogous to HGPRT. And one of the things that it does is maintain balanced nucleotide pools. And so if you're lacking PNP, you get an imbalance in your nucleotide pools. And when your cells go to rapidly synthesize DNA in the S phase of the cell cycle, then it doesn't proceed efficiently, and you often get an S phase block. So this is particularly important for example, for lymphocytes, leukocytes that have to expand rapidly, then this can really impact them, and hence the relative immunodeficiency. So interesting, definitely not what we expected to get out of an interferon screen, but we thought let's go ahead and check it out. The structural diagrams weren't very striking. So in general, both when we tried to structurally model the polymorphism or if we looked at some of the public databases where you can test whether polymorphism is likely to be damaging or not damaging, it would not predict it to be very disruptive. And I guess that's what we figure. This is a pretty common polymorphism. So common things are typically not that highly disruptive to gene function or structure. But we could see some effects. So the top panel is looking at B lymphoblastoid lines. We noticed that there was a decrease in mRNA expression if you carried the risk allele, which is in red. Green is the homozygous non-risk, and then intermediate is the blue heterozygote. We did see that there was a decrease in the protein level that corresponded to the transcript level. And then a nice thing about this enzyme is that it's been studied a decent amount, and there's actually a colorimetric assay that had been published already. So we were able to test enzymatic function and normalize for the amount of protein, and then see if the enzyme functions similarly if you could control the amount of protein, and it didn't actually. So there was a decrement in the enzymatic function here, and this is when you sort of have normalized so that this quantitative difference is not there anymore. So it seemed like we were dealing with an allele that likely had an enhancer or some other variation in the promoter where it was slightly less efficient, and then also that likely the protein coding change was impacting enzymatic activity. And so we saw similar things in the lupus patients when we looked at B cells for the transcriptional and protein level. Looking at enzymatic function, you need a lot of cells. So we actually did red blood cells because it's highly expressed in red blood cells as well. And you could see a similar trend, at least with the risk allele carriage. It was hard to get any primary cell population that we could get in large enough amount to do the colorimetric assay. Okay, so looking at cell cycle, sorry, this is a little bit busy, but I'll take you through. So this is the sort of typical cell cycle plot with S phase here. At rests, these B lymphoblastoid cells didn't show much of a difference based on either the CC non-risk or the TT homozygous risk genotype. But again, because of the pathway knowledge, we knew that there were ways you could provoke it and make any defect worse by adding more of what the enzyme already has too much of. So if you add deoxyguanosine, you could see S phase block, and this was actually more pronounced in the TT genotype. If you really crank up the deoxyguanosine, then you can block cell cycle and S phase in quite a few of the cells, and you sort of lose the genotype effect again. So this is illustrated in B, much more easy to read and more graphically. At the intermediate concentration, you could see the genotypes playing out where you had a greater S phase block in the risk genotype, intermediate in the heterozygote, and then least in the homozygote. And then a nice thing is that with the knowledge of the pathway, you can actually bypass it with hypoxanthine. And so we're able to show that you could actually put in a pretty large amount of deoxyguanosine and see normalization, and you could rescue the phenotype with hypoxanthine. So that just helped us feel like we're on the right track and we weren't mistaking ourselves with the gene variant being responsible for what we're seeing. Okay, so pretty weird, right? So a cell cycle, but we found it based on interferon. If we looked at panel A is just resting lymphoblastoid cells and looking at their interferon-induced gene expression, you could see just in resting culture that there was a greater interferon-induced gene expression and the risk genotype as compared to non-risk genotype. This seemed to be consistent. It wasn't necessarily a stimulated effect, so we tried some interferon stimulations, and really there's a separation at the start, and it maintains fairly parallel separation throughout different levels of stimulation, suggesting that it's there at the start and it persists, this difference. We were wondering if PMP might be induced by interferons, and it didn't seem like it. These are pretty flat lines actually with interferon stimulation, maybe slight induction or maybe not. Similar phenomena in lupus patient B cells, and again, PMP wasn't induced in those cells either. So we thought about this for a while, and we thought, the B lymphoblastoid cell lines, they can make a little bit of interferon beta, but not a lot. And we were wondering if there might be some intrinsic effect. And I'll say one of the things that makes us think about that is that when we've done our interferon studies in patients, we've noticed that if you measure functional interferon in serum, and then you take cells from that exact same blood sample, same day, and you do sort of a classical interferon signature in those white blood cells, there's a pretty good correlation. So it's shown in the graph. We correlate pretty well, but the typical finding is that there's a skew and that there are more patients that have high levels of interferon-induced gene expression than have high levels of circulating interferon. And you can find some that basically don't have much of an increase at all in circulating interferon, but do have an interferon signature. And so it's interesting, I think and that's a pie graph just trying to simplify it a little bit, that we think there's this group that's sort of signature positive, but doesn't really have any detectable circulating functional interferon activity. So, we believe this group, at least in part, might be due to more of the intercellular receptors. So things like MDA5 or IFIT1, RIG-I, possibly STING as well. There are ways you can turn on these interferon-induced genes, of course, without triggering the type 1 interferon receptor. I think we're a little bit at risk of hypnotizing ourselves by calling them interferon-induced genes, because they are induced by interferon, but they're also induced by other things. And they're probably might be better to call them antiviral response genes, because it's not just only the type 1 interferon receptor that can induce these genes. So we thought about this, and there's some interesting literature in the cancer field, actually, that sort of put me onto this, and this is where the project stands and where we're going. So in the cancer literature, there's a description of an interferon signature that's induced by some of their chemotherapeutic drugs. And these drugs, many of them induce an S-phase block as their mechanism to just kind of go after rapidly dividing cells. So the idea is just, these are the old school chemotherapeutics where you just kill rapidly dividing cells, and of course cause a lot of bad side effects as well. But they noted that you would get formation of micronuclei, which are these fairly large cytosolic DNA inclusions, and that they were correlated with the interferon signature in the tissue. And so these happen during S-phase block. Presumably, they're abortive chromosomal replications where things get blocked so much it doesn't work, and the nucleus ejects them. In the cancer literature also, it was found that these could signal through STING, and so gave us quite a lead. And so we've been working on studies here. So in the pictures, this is a picture of a cell that has these micronuclei. So there's a nuclear stain, and then this is green is picogreen, and then there's sort of a combination of the mitotracker, and you can see the sort of outline of the cell, and you can see these punctate discrete inclusions that are, again, fairly large because we can visualize them on the microscope. So these aren't little oligonucleotides, these are large clumps of DNA in the cytosol. And we found, if we study this, that this tended to be an all or nothing phenomenon in the cells, which was kind of lucky for us. So we could enumerate them as a percent of cells that have micronuclei versus ones that don't. And we could see that this was fairly greatly increased, more than double in our risk genotype versus our non-risk genotype. So we're moving ahead with some of that in the human sphere, trying to document this a little bit better, and also to try to determine which way this might be signaling to induce interferon, whether this is STING or TLRs or both. And this is our first example where we've actually taken a gene back into a mouse. So we made the PMP flox mouse. Humans only have one PMP gene, but mice have PMP1 and 2. It's unclear how much each one contributes. So they're right next to each other, and we just got them both with the same model. And we've done induced models so far. We've done pristane and imiquimod, and we're just sacking the mice now. And I'm very curious to see if this makes the lupus phenotype worse, and if so, in which ways. PMP is very ubiquitously expressed. So that was another push to sort of make the mouse model, is that we want to get sort of an overall in vivo phenotype, and then we can go back with specific Cres, to try to explore some of the more striking findings. So we're hopeful. The student working on this is very hopeful. If there's no phenotype, that's not so good, but we'll see. And hopefully it'll be a nice model for us to study this, and to also do some of the micronuclei follow-up in the mouse as well. So, okay, so this is my last slide here, that we have noted this PMP variant that's associated with interferon and decreased enzyme expression and the cell cycle abnormalities. And so we're interested that this, these micronuclei could be a novel mechanism for sort of a cell intrinsic interferon pathway activation, that this could maybe happen. And maybe this, in those lymphoblastoid cells, maybe they make micronuclei, and then that could stimulate STING and interferon induced genes directly without so much external interferon secretion. So, okay, well, thank you. The list here is all these different people who've contributed to the work through the years, and it's been a great laboratory group, and I should show you. This was the best we could do. We got almost everybody in person and one person on Zoom, so we took the picture. And it seems like it's rare that we can ever get quite everyone in the same room at one time, so as close as we could get. And thank you very much for your attention this morning.
Okay, thank you very much. This was a very fascinating seminar. I think we have a number of questions, but I'm gonna start with one of my own just because I don't want to forget it. The micronuclei, uh, have they been DNA sequenced to determine that they really contain chromosomal DNA? Because they could contain, in principle, other kinds of DNA like mitochondrial or even reverse transcribed nucleotides.
Yeah, that's a great question. I don't think that they have. One thing is that they do carry with them a nuclear membrane. And so that's very suggestive. And that's one of the stains that we're doing now to try to confirm. People that are into this field had told us, that you should document a nuclear membrane around them to really prove that they're micronuclei. So the presumption is that that it's nuclear instead of mitochondrial, for example. But I don't know. I mean, it's always good to check these things. And yeah, I imagine if you lyse cells, you should be able to purify you should be able to find a way to pull these out, being sort of smaller collections of DNA. So um, I'm not aware, but that doesn't mean it hasn't been done. I should take a look. But yeah, I'm not aware of studies where it's been sequenced.
Yeah, thanks. So we have a question from Rihanna Jar. Are the studies of anifrolumab efficacy stratified by interferon risk alleles?
So I'm not aware of that. And in general, it seems like a lot of companies have not at least talked about or definitely shown in publications much genetics anymore. I've since kind of a decreased enthusiasm for, GWASing every patient that they get in their clinical trials. I think the best thing you've got there is looking at the signature as they've done. And unfortunately, in the phase 3, not really seeing a large difference between, with the signature being able to predict response or non-response. And so I think it's too bad because I think the signature has a good amount of non-specificity to it. You get some of the same genes induced by interferon gamma. So and potentially, as we mentioned, by cell intrinsic, phenomena that may not be impacted by anifrolumab. So some additional detail there would be interesting, whether it's genetics or, looking at other, cellular programs. But I'm not aware of any good data about that.
Yeah. So another question from Dr. Najjar. Besides the interferon risk alleles, are there other known causes of high interferon in lupus?
I mean, the strongest factor, which I didn't dwell upon that much in this talk, but the autoantibody specificities, if we take all the clinical features we've got and put them into a big regression, the autoantibody specificities come out at the top. So we think that that's probably a direct effect, that they're making immune complexes and then being immunostimulatory. And in fact, we think it probably confounds some of the studies where you look at SLEDAI, for example, and having double-strand DNA as a component of the SLEDAI, and then you see a correlation with interferon. And you think, okay, but maybe that's because one of the factors in the SLEDAI is a direct inducer of interferon, so maybe it's not fair. You do see, I mean, that's an easy thing to try, is if you just drop out double-strand DNA, there is still a correlation. So the whole thing doesn't go away when you take away DNA, but it's definitely a factor. So yeah, after that age, I mean, age is interesting because when we've studied age and interferon levels, again, this is in circulation is the best thing we've got. But in healthy people, you see that younger people have higher circulating interferon levels than older people. Same in lupus relatives. And so there's sort of a generalized age trend that seems to be independent of lupus, but the levels are much more exacerbated in lupus, and it's easier to see, I think, in the patients.
Okay, we have a question from Mark Wenner. Great talk, thank you. Is the RF5 association with anti-SSA row linked to row 52 or row 60 or both? Are there different genetic drivers risk alleles for interferon signature for row 52 versus row 60 in SLE patients?
That's great. We haven't looked, and so I don't know actually. So we haven't had that level of data available to us in any of the large cohorts. And this is probably, I would say, a general defect in the genetics cohorts, is that the larger numbers with less detail is almost always the way it goes. And that's the way people collect them. And so I haven't been able to look at row fine specificity. I guess one slightly related thing, we were able to look in anti-Rho positive mothers of neonatal lupus pregnancy. And that was interesting in that people who just had anti-Rho and nothing else at all tended to not have high interferon levels, whereas people who had some symptoms tended to have high interferon levels. And so I think that may be getting at whether you have enough background alleles to sort of, make the synergy or not, So, but yeah, I don't know about those fine specificities, unfortunately.
Then we have a question from Keith Elcon. Uh, Micronuclei are relatively easy to see. If you look in 10 SLE patients, PBMC, how many have micronuclei?
So you mean, how many have at all?
Yeah.
Or yeah, I think we often will see some in some patients. So it's often if when we look, we can see some, and I think that's likely an impact of cellular stress and other things. So we're still doing these studies, but so far it looks like we're getting more micronuclei again in the context of the risk versus non-risk, but the non-risk is not zero. So we still do see micronuclei in some cells, even irrespective of this particular genetic variant that we're looking at.
Do you have any hypothesis for how they form?
It's a hypothesis, in that potentially defects in cellular replication or other stressors, that might impact cell division. That's the connection between the S phase block. At least that's the hypothesis. Again, I don't know if that's the whole story, but this idea that you have some sort of an abortive chromosomal start that then gets blocked, and then for some reason there are enough errors or something else that it can't go forward. But that's a fairly hand waving, I'll admit.
Yeah. Are they in any particular immune cell lineage?
We've looked mostly at B cells, although that's out of convenience. And the convenience is that we have these large libraries of lymphoblastoid cell lines that have been made from the genetic consortia. And so you can order, 5 or 10 cells of one genotype and know it's true in 5 or 10 of the other. With a lot of our genetic variants, especially getting homozygotes, it's a small portion of the population. So even with our lupus patients, we have a pre-genotype cohort we draw from, and then we've been doing B cells just to line up with the lymphoblastoid cell data. But it's a good question. We are curious to at least try to look at some other major lineages, like, at least CD4, CD8 T cells and things like that. But we haven't gotten to do that yet.
Yeah. Okay. Thank you. We have one more question, running short on time here, but from Mark Wenner. Is it known if SLE anti-DNA serum binds micronuclei and relative avidity compared with nuclear binding?
That is not known. It's a great question. Again, getting at this, what are the source of the nucleotide, triggers in lupus, and yeah, not known. And there's not too much about micronuclei yet in lupus. So we're hopeful to explore and see what we can find at it. That's still probably a little ways away from what we might try in the next few steps, but it's a great question, really fascinating for sure.
Yeah. Okay, those are the questions we have at the moment, and
Okay.
I think people are moving on to other meetings that start at 9 o'clock. So